So next speak is Dr. Jinyue Liu. He's a young scientist from Genome Institute, Singapore. Her lab developed nucleic acid based on technology to investigate the human brain development function and the disease. Please, Dr. Liu.
Hello, can you hear me?
Yeah, we can hear you.
Yeah, hi. Thanks, Professor T, for the introduction. So I'm Jinyue, and I lead the Laboratory of Single-Cell Spatial Neuromics at Genome Institute of Singapore. And today I'd like to share a story on how we harness the Human Brain Atlas and the organoid model to understand human brain development and disease. So this is a work in preparation, and we welcome any comments and feedback. So this audience really needs no introduction to organoids, right? I think we all agree that organoids are an essential complement to the human cell atlas, and rightfully so, because unlike primary human tissue, organoids are sustainable. They are a renewable source of mini organs, if you like. And because they are derived from human cells, they are truly human, and they are capable of recapitulating human-specific traits. And that is particularly true for the human brain, and I will go on to describe some of them later on. And a nice thing about the organoids being an in vitro system is that they are experimentally amenable. And that means that you can manipulate them and track changes over time. And this is particularly useful for developmental studies. Last but not least, while we work towards a consensus human cell atlas, let's not forget that each of us are unique individuals, and organoids are fully capable of capturing such individuality. And that's the basis to personalized medicine. I'm a neuroscientist, and in line with my long-standing interest in neurodevelopment, I'm leveraging the brain organoid system to study a condition called autism. So autism is a chronic psychiatric disorder with developmental origins. And patients with autism, they struggle with social communication, repetitive behaviors, restricted interests. And some of these symptoms could be self-injurious or debilitating. And they are definitely a huge source of stress for their caregivers. And mitigating these symptoms have been challenging because autism exists on a spectrum in both its causative roots as well as the manifestation of the symptoms. So we actually know a lot about the genetic influences behind autism. So here showing you on the left is a circular map of the human genome from the Safari website. And each of these bars represent a gene that has been implicated in autism. And you can see that there are more than 100 different genes that are associated with autism. And even up to today, as you can see on the study on the right, there are still many genes being found through exome sequencing. So we do get some clues from many pioneering work that other researchers have done. And this is some clever co-expression network analysis where they pinpointed deep cortical neurons in the mid-fetal stage to be a key node in autism biology. With the advent of single-cell technologies, more and more labs are starting to look at cell type-specific gene expression changes. This is one of the more recent ones where from Arnold Krestin's group, where they look at post-mortem brains from autistic subjects. And you can see that in these, on this plot, several significant genes being highlighted and colored by the cell type of enrichment. And you can see that many of these genes are present in the upper layer neurons. So, how do we interpret these predictions, and how do we know how the gene expression changes affect the cells, right? We know a lot about all these genetic influences and the transcriptomic changes, but the emergent cellular phenotypes, right, which is the link between the genes and behavior, is still relatively unknown. And so that's what we set out to understand. We don't have access to human fetal brain samples, so we model our study in human brain organoids. And we have 5 cases and 5 controls. And we take a 2-pronged approach to map both the spatial landscape, meaning the tissue architecture, as well as the molecular landscape, meaning the molecular expression of these individual cells. And we're interested in the early stage of the brain development for several reasons. One, because high confidence autism genes are generally expressed over fetal brain development, and as you can see here, somewhere between 10 to 18 post-conception weeks. And over this time period, a lot of interesting things are happening to the human brain. You get formation of layers, cortical lamination, addition of layers, expansion, and so on and so forth. And so this time period also corresponds nicely to the stage of human brain development that organoids could recapitulate very well. So just as for any organoid study, we benchmark our systems extensively. What you're looking at here is the lifespan of an organoid in our hands, starting all the way from an induced pluripotent stem cell colony to an organoid at 8 weeks. And you can see these protrusions from the organoids. Each of these protrusion is what we call a neuroepithelial bud. You can think of each bud as a tiny unit of the brain. And if we take these organoids and we dissociate them and do single-cell RNA-seq, we find that we are able to recapitulate many, or in fact most of the canonical cell types that you expect to see in a human fetal cortex. And each of these cell types express the predicted as well as novel markers that we know of. And importantly, when we compare the transcriptome to the primary tissue, to human fetal brain tissue, we find that the transcriptomic similarity is strongest to the stage of development that we're interested in, which is between the 12 to 13 week range. So in addition to cellular composition, we also check for cellular organization. And this is important in autism because there have been studies showing that the laminar organization of the cortex might be disrupted in some cases of autism. And what you're looking at here are images of brain organoid sections stained with several protein markers that represent different cell types. And you can see this very nice lamination pattern and the orderly arrangement of the different cell types in our organoid system. So we know that our organoids are reliable. So having benchmarked our organoid system, the next question is, are our organoids good disease models? And to answer this question, we look for the growth signatures of the autistic human brain in our autistic organoids. First off, just comparing cell type composition, we see that our case organoids do show the complement of cell types that the control has. So these are just a UMAP representation of the single cells obtained in our single-cell RNA-seq experiment. There's no integration, just a simple merging of gene cell matrices, and you can see that the cell types align together very well. And another phenotype that we observed in our organoids is that the neuroepithelial buds that I was describing earlier on, they tend to be bigger in size in our cases, and that's quantified over here. And this corresponds to, it is very reminiscent of what we see in a proportion of human autistic subjects, where there's an enlargement of the brain or the head circumference. And finally, we see an upregulation of FOXG1 expression. So this is a forebrain marker, and yes, that has been previously reported to be associated with autism. We see that it's upregulation in our case organoids as well. So having shown that our organoids are a reliable model of the human brain and of the human cortex to be specific, and they are capable of modeling the disease, we then first ask, what are the obvious differences between our case and control organoids? And one of the differences that jumped out was the cell composition, the proportion of the major cell classes in our organoids. So granted, we do see some sample-specific variation, but overall, we see an enrichment of neurons in our cases, as opposed to our controls. Here's a different view of the same results, again showing the depletion of progenitors and the enrichment of neurons in our cases. And if we take a deep dive into each of these individual classes, we begin to see that there are specific cellular subtypes that are affected in cellular composition. So this initial observation raised a lot of questions. So for example, where are the extra neurons located in space? How do they fit into the tissue architecture? What are the cell-cell signaling that are perturbed? And to answer these questions, we turn to spatial omics. This audience don't need an introduction to spatial transcriptomics. In fact, there's a concurrent session happening right now on spatial transcriptomics. So I'll just give a simple analogy to bring the point across. So imagine you're a sailor lost at sea, and all you see in the night sky is just single star. Well, you need a lot of luck finding your way home. But if the night sky is full of stars that night, well, you can use this inbuilt compass to navigate and find your way home. So this is the exact same concept that we're applying to our spatial omics studies. By knowing how cells are positioned relative to each other, we can begin to identify the abnormal cellular events and leverage knowledge of these events to improve our understanding of autism neurobiology. So the spatial technique that we are using is what we call multiplexed fish or merfish, similar to SIGFISH that Long Kai described yesterday. And this is how our data looks like. What you're looking at here is just a panel of, it's one of the butts in a cross-section of the entire brain organoid. And here are 7 genes out of 94 genes in our panel. And you can see that you are already getting, these awesome layers with just 7 out of 94 genes. So then the question becomes, how do we make use of this data to inform the neurobiology of autism? So what I'm going to do for the remaining time is to describe some of the approaches that we have been exploring to get at this question. So we are interested in understanding how the tissue domains are being mapped because we get layers in our cortical organoids, right? So this is a very relevant question. Are the layers changed in any way? We are also interested in the cell type distributions because, as we know, the location of the cells would change their function and the development of the brain. We are also interested in the interactions that happen between the individual cells. And so to answer all of these questions, we have been exploring a variety of approaches, and I'll just go through some of them. So the first was the attempt to go single cell. And in this aspect, we came across a very surprising challenge, which is that the cells in our brain organoid system are actually very densely packed. So you can't segment them using conventional methods. Thankfully, lucky for us, there was a deep learning method for cell segmentation that was recently published. And using a modified form of this method, we were able to segment cells in the brain organoid very well. It does as good as the human eye. So going single cell is a game changer because it now means that we can apply a lot of the single cell techniques that we know about to this spatial data. And one idea was, to ask how the individual transcriptomic cell types map onto the tissue space. And so here's an example of how we do that. Here's showing you 3 genes that we have probed for by multiplexed fish. And if we do this integrative analysis, we can predict the cells that express each of these genes. And here are using 3 canonical genes that we know about, and you can see that they match up very well. And now that we are able to do this sort of integrative analysis, we can begin to impute genes that are not originally present in our probe library onto the same tissue map space. So on this first row, what you see are the measurements from our multiplex fish experiment. And if you take each of these gene, if you remove each of these gene from the integration process and make a prediction on where they would be, you can see that the prediction matches the measurement very well. So now then, with these approaches, we are able to find where transcriptomic cell types are in space. And we are starting to find that the transcriptomically identified cell types are perhaps sharing different spatial distributions in organoids from different patients. And we would like to be able to map the differential genes in space and start identifying the cell-cell interactions that are responsible for the altered cell type composition that we saw earlier. And with that, I'll end by thanking a wonderful team. This is the result of a huge collaboration. OneKey generated a lot of the brain organoids and single-cell RNA-seq data that you saw, along with a team of very fearless warriors and interns. Sham was instrumental in the data analytics aspect, and we got a lot of support from Kok Hao and his team on the MERFISH or the multiplexed fish implementation and analysis, as well as our partners from Bioinformatics Institute, and our funding sources. And I will stop there and take questions.
Thank you. Yes, you have one question. Did you say your question?
Let me see.
Let's say the Autism is a human-specific disease. What's the cell source to build Autism brain organoids?
Yeah, so that's interesting. So I guess the question is, if autism is a human-specific disease, how do we model a behavioral disease? Is that the question, I'm guessing? Well, I will guess that that's the question. So the cell source that we use to build our organoids are essentially commercial. So there are many stem cell banks out there that actually collect cells from these autism patients. And some of them are very clear genetic mutations. And so that could be a form of, that could be a source for generating these autistic brain organoids. I hope that answers your question.
Thank you. I don't know the Dr. Julia, did you have another questions? Anyone has another question?
Okay, I see a question from Jay. So Jay, you're asking how many genes for MERFISH are sufficient to impute spatial gene expression? Yeah, so our libraries are pretty modest. So we typically stain just like 100 or hundreds of genes. And that was the example that I showed earlier. So I would say that 100 is probably good enough, or even less. And if you, I think people have tried like something like 250 genes, and that is sufficient to fully to accurately perform the imputation. Yep, no problem.
So thank you.
Okay.
So no more questions. We thank you, Doc, to give an excellent talk. Thank you.
Thank you.